Okay, so today we are going to be starting on a new pair of units. So we are going to be working on these kind of together. So the first half of the week we're going to be doing mitosis. Second half of the week we're going to be doing meiosis. So two different processes, both of which are going to explain how the cell reproduces. So everything that we've talked about so far, all of the organelles, all of that, have been leading up to kind of DNA replication and cell replication. So this week we're going to do how the cell divides, and next week we are going to do stuff with translation and transcription, which is very fun if you like puzzles. But yeah, that's what we're looking at kind of over the next 2 weeks. And then after that is Thanksgiving break. Yep. We've got 2 weeks left. So, first thing, the main thing we're going to be focusing on in this unit is kind of how different processes in the cell are vital to not only creating healthy organisms, but also to provide uniqueness. So all of you look around the room, all of you look different, and that is because of, what?
We're twins.
Not quite. Even identical twins have some difference to them. But how our uniqueness is based on how our DNA is moved and how our cells kind of combine and reproduce and that kind of thing. You guys in the back. So the first thing I want you to do, take a couple minutes, write down on your paper, how would you get from the picture on the left to the picture on the right? Take a couple minutes. So, anybody think they know how we could get from DNA number 1 to DNA number 2? Peter?
More DNA strands.
So we are. So you did notice that in this one we added, guys in the back. Don't make me move you forward. So you did notice that there was 2 strands over here. So you can see there is a blue strand on one side and there is a yellow strand on the other side. So maybe it has to do with adding more DNA. Any other ideas?
Didn't you say we were like switching at like the marking period?
Potentially.
Shut up.
So we could do mitosis.
I like the family.
We're not doing photosynthesis here. Okay.
Mitosis.
We are, yes. So this would happen in mitosis.
It's the whole
So,
point of the
yes,
lesson.
yes. So this is what we're going to be talking about today. We are going to be replicating our DNA, because in order to divide, to create multiple cells, we need to have multiple copies of instructions.
Isn't that how you like know people?
We'll talk about that. Yes. So, the basics of cell replication, or DNA replication in this case. Yeah, I don't know why that's cell. DNA replication is that this is going to occur in the nucleus. So we have bounced around a bunch of different organelles. We've bounced around the mitochondria, the chloroplast, the cell membrane, the lysosome, the Golgi, all of those fun ones. Now we're hitting the nucleus. So the nucleus is going to be the center for genetic information. It's going to be where all of your instructions are stored. So if I grab from over here one of these next booklets over here.
Oh.
Yes, kind of like Legos. But this would be kind of the instructions that are stored in your cells. All of the instructions on how to build your cells, build different proteins, control different functions, control different genes. All of those are stored within the nucleus. And this process of DNA replication is going to occur in a stage called interphase. So we kind of look at cell or DNA replication and cell replication as kind of a calendar. So you can see down here on the bottom we have this kind of circular, this bit right here, this tiny pink sliver, is our actual cell division. So it only takes up a tiny bit of the cell's life. The rest of it is devoted to growth, to copying its DNA, and more growth. So you can see there's a G1, an S, and a G2 phase. Those G1s and G2s are just devoted to getting bigger. So eating molecules, getting larger in size, doing all of the things you normally need to do. This blue one here is just devoted to DNA synthesis. The cell is going to copy all of its things, or all of its DNA. So if I told one of you to go to the copy machine, make 10 copies of the test from today, same thing. And this is going to create new copies of the DNA for each daughter cell, so that each cell that is created from mitosis has the same instructions so they can do the same things. And this is going to occur or be performed by multiple different specialized enzymes. So we're going to talk about those enzymes. I'm going to go a little more in depth in here, but we will be talking about these different enzymes. This is a big process. We are going to model it, hopefully today, assuming we have time. If not, that'll be the first thing we do tomorrow. Any questions about this? There we go. So, our DNA replication is going to look very messy. There is actual organization to this. So we kind of take it in different steps. So the first thing we do, Gideon, the first thing that we have to do when we are looking at our DNA, and I have fallen in love with this little model. Yeah. So this is our DNA. So we have it in what's called a double helix. If you remember, the double helix is the ladder formation. So you can see that it looks like kind of a swirly ladder. So in order to copy our DNA, think, how would you copy the information from the person sitting next to you? Hopefully none of you would have.
You tell them to lift the bird out the window and then you steal the paper.
Okay, but what do you have to do from their paper? You have to write it down. So in our case, we are going to use our partner's copy as a template for our own copy. It'd be like I said, if you just cheated on the test, you have to use their answers to fill out your paper. So the first thing that we are going to have to do, so we need to split it open. So we're going to use an enzyme called helicase. One thing you'll notice with all of these helicase, polymerase, ligase isn't on here. If you see an ASE at the end of a word, probably going to be an enzyme. So our first enzyme, this helicase, its job is to go through like a zipper and unzip our DNA. So we have to physically pull it apart, because whenever we're doing it like this, you can't copy the bases if it's still stuck together. So we have to unzip it, kind of like a jacket, in order to access the different nucleotides. What are nucleotides again?
It's like the things in the nucleus. Or outside of the nucleus.
Not quite.
It's the monomer.
It's the monomer of a nucleic acid. Our nucleic acid, DNA. Again, all of this stuff we've covered before. But we are going to unzip our DNA. And from there, we have these little primer enzymes, these primase, which aren't on here, are going to stick a little piece of RNA, and that's going to tell the DNA polymerase, this big yellow thing, where it needs to go. It's like if I give you a set of instructions on go to lab bench 1 and make the models, the telling you where to go would be the same thing that primase does. The DNA polymerase is then going to attach it, and it's going to take these free-floating nucleotides, and it's going to keep sticking it onto the DNA. It's going to do this in a very special direction, which we'll talk about in the next slide. It's very specific on how it does it. So a DNA polymerase will stick onto the top, it will stick onto the bottom, and it will start coding in a special direction. From there, it starts to create our new DNA. So we start towards the end, work our way up, and that's how we are going to create 2 copies of our DNA. So like I said, it's kind of like cheating if you use your neighbor's thing. Each of these different pairs, anybody remember the 4 letters that we use? A, T, C, G. A always bonds with T, C always bonds with G. So in this case, the polymerase can figure out that, oh, an A is there, I need a T. A C is there, I need a G. A G is there, I need a C. And it just keeps stacking them on. If it messes this up, that's when we get mutations and things like that. But that'll be next unit. Yes. Any questions about this? Oh. Okay. So the next thing I want you to do is on your own, so I should not hear anything. And then we'll discuss it. Define these 2 terms, lead and lag. Brayden, L, what does it mean to lead?
I said that's like the first strand to separate from it.
Okay, so just generally, what does it mean to lead?
To go first. To be in first place.
To be better than everybody else.
Okay, to go first. To lead, usually you're the first person in line, you're the first person that everyone else is following. Kyle, what does it mean to lag?
I said be slow down, maybe lag. I am add to that.
What else, Noah?
So you're playing Minecraft, right?
Oh, let's borrow. No, it's
I'll just borrow the pen. Yeah.
You're on
Alright.
a really old iPad.
The only person I should hear is Noah.
You're on a really old iPad, right? And you just blow up 75,000 TNT.
Okay.
And then you just start lagging, you know?
Okay.
So you're going really slow. Yeah.
Okay, so can't move, slow, falling behind. Okay. So, then if we talk about the leading and lagging strands, these are going to be the 2 sides of our DNA whenever we split it open. So the leading strand is going to build continuously towards the replication fork. Usually this is from the 3' end to the 5' end. What do I mean by 3' and 5'? Well, if I actually grab from way back when, anyone remember these?
Oh, yeah.
The little molecules that you made? So, whenever we talked about DNA and RNA, I told you to make these fit the best way possible, and they happen to fit upside down. That's what I mean by 3' and 5'. So whenever we talk about actual DNA, it sits kind of like if I had one of you stand on your hands and be held up by another person. No, we're not actually doing that. I don't have mats or anything. No. So if one of you was standing here normally, the other person was standing on the opposite side doing a handstand, that's kind of how DNA aligns itself. One side is going to have a 3' carbon, one is going to have a 5' carbon, and they kind of run anti-parallel is what it's called. So a normal leading strand runs exactly like that. It runs continuously, it doesn't have to stop. But you notice, if we have to do it on the other side, if this DNA polymerase on the top runs straight towards the replication fork, the other one, if it can only run from 3' to 5', it's going to run backwards. It's going to run away from the replication fork. And what happens is, the farther this gets out, if we start our polymerase here and go over, we still have this whole section that needs to be redone. And that's what I mean by lagging. So your analogy with the video games is actually kind of right. If you think of if you've lagged in a video game, it's usually really choppy, really like sputtery.
That's what happens when you build too much on the Nintendo Switch.
Okay. But in this case, the lagging strand is going to have to be built in segments. So these are called Okazaki fragments.
Is that Japanese?
It's named after a researcher who is Japanese, I believe, yes. But this is going to be moving away. Guys, this is going to move away from the fork, and it's going to have to be made in sections. So the DNA polymerase will make this section, have to jump up further, make this section, and it's built in fragments, in choppy bits. But it's still going to come out the same way that this top one will come out. And these segments are going to be connected by a thing called ligase. Again, ligase, ASC, is an enzyme that connects the different fragments. Any questions about this? So we're also going to say that our DNA replication is called semi-conservative replication. So what does it mean to conserve something? So to keep it. So if you conserve something, think. No. What does it mean to conserve something?
Specifically, what?
What's it mean to conserve something?
Conserve?
Yes.
So to conserve is to like, how do I put this?
Yeah.
It's to like, kind of, yeah, like use wisely and like not just like overuse something, but kind of like savor it.
Okay.
Save it.
So to save it. So semi-conservative means that we're going to keep a part of it. So whenever we replicate our DNA, each copy is going to have half of the original DNA.
Yeah.
So it's going to do this through semi-conservative replication. So the daughter, or both daughters, are going to have at least one identical copy of each chromosome, or of each set of DNA. So when we talk about, like I said, semi-conservative replication, each of the parent strands, or half of the parent strand, is going to go to each of the daughter cells. So this is going to serve as the template, like I said, if you were cheating off of someone, copying the DNA strand into the new strand. And like I said, complementary base pairing is going to help control and assure that the newly made strand is going to be identical to the one that got removed.
Complementary, doesn't that mean like the two angles add up to a 90 degrees?
Okay. Yes. But in this case,
But we're not in geometry.
so to complement something is to be kind of the other half. So, yes, in your case, two halves of your two angles add up to equal the full 180. In this, half of the strand, for complementary, we're always going to pair A with T. We're always going to pair C with G. So that is something you'll want to remember, especially for this unit and especially for next unit, because next unit we're going to be applying all of that and making our own proteins. Yes. Any questions about being semi-conservative? Any questions about DNA replication? Anything like that? So this is going to be applied when we talk about mitosis and meiosis, because we need to do this in order to be able to replicate our cells. So we have to have one copy of our DNA, at least one copy, in all of our cells, otherwise we can't function properly. So this is going to be very important going forward when we talk about mitosis and meiosis and all of that fun stuff, because this is going to control how our cell functions.
Can we die?
if you are missing DNA, if you are missing chromosomes, can lead to deformities, can lead to diseases, can lead to all sorts of fun things. Yes, we'll talk about mutations more next week. This week, however, we will be talking about one disease that does influence how replication occurs. We will talk about that towards the end of the week.
Okay.
Yes, there will be some medical applications to a lot of this. Okay, so as you finish up, I have a short video for us.
Amoeba, sister.
What?
Amoeba,